So just what is gut taste? And who wants to taste the contents of the gut, already halfway from food to feces? Taste is often used to describe overall sensory perception of food. However, from a scientific standpoint, taste is specific to sensory stimulation in the oral cavity. This distinguishes taste from flavor, which includes the textural and odor components of food. Most flavor is actually perceived through retronasal olfaction. Taste includes only 5 specific sensory sensations, generally agreed upon by the scientific community: sweet, sour, salty, bitter, and umami. However, other tastes may also exist, such as starch, fat, various minerals, and carbon dioxide. There is no universal agreement on the definition of taste, even in the oral cavity. Some basic criteria for the so-called primary tastes could be as follows. 1. The taste must provide a survival advantage. For example, detecting sweet sugars or umami proteins helps organisms identify sources of energy and protein. 2. There should be a well-defined class of effective ligands for the taste receptors, such as acids for sour or proteins for umami. This criterion becomes tricky when considering tastes like sweet, which has a wide variety of chemical structures that serve as ligands. 3. The ligands must stimulate unique transduction events. In other words, taste should not include stimulation by other senses, such as chemical irritation. 4. The signal must be conveyed by gustatory nerves. 5. The stimuli must generate unique sensory qualities. In other words, aspartame and sucralose are both sweet, so they should not be considered separate tastes. 6. The stimuli must evoke a physiological response. For example, the sweet taste of sugar can result in a small insulin response. All generally accepted taste qualities do not meet these criteria perfectly, yet these rules provide some basis for the classification of sensations as tastes rather than other modalities. Taste in the oral cavity is detected by taste cells organized into taste buds on papillae. These taste buds are onion-shaped clusters of cells with a pore near the top, which allows tastants to interact with the taste receptor protein. Taste receptors for sweet, umami, and bitter are G-coupled protein receptors. The receptors span the cell membrane and are bound to the G proteins in the cytosol. Activation of the extracellular domain results in the cytosolic portion releasing a subunit of the G protein. In many taste receptors, this protein is alpha-gusteucin. These G proteins then initiate a signal that leads to the sensation of taste. The G-coupled protein receptors involved in sweet and umami are composed of 2 subunits of taste receptor type 1, abbreviated T1R, members 1, 2, and 3. For sweet, the subunits are T1R2 and T1R3, and for umami, the subunits are T1R1 and T1R3. Bitter receptors contain only 1 unit of the T2R family. Over 30 proteins of this family have been identified as unique bitter receptors. Salty and sour tastes are believed to rely on ion detection, with salty most likely mediated by epithelial sodium ion channels, or ENaC. When these receptors are activated by compounds, the taste cell depolarizes and elicits an action potential. Many different receptors have been implicated in sour taste, including acid-sensing ion channels, hyperpolarization-activated ion channels, 2-pore domain potassium channels, transient receptor potential cation channel subfamily P members, including PKD2L1 and PKD1L3, and apical proton conductance-mediating molecules. Again, the flux of ions into the cells mediates the response that we identify as sour. As taste receptors are proteins, it makes sense that the human body would allow the same protein to serve different functions throughout the body. In fact, some of the same receptors involved in oral taste perception can also be found in other parts of the body, including the kidneys, lungs, sperm, and as we will discuss in this presentation, the intestine. So if these receptors have important biological functions, how does receptor activation yield a response? There are both nervous and endocrine pathways by which gut taste may elicit a response. Nervous stimulation, once initiated in the enterocyte, can be carried by the vagal afferent neurons to ganglia of the spinal cord or to the nucleus of the solitary tract in the brain. A response impulse is quickly dispatched back to the enterocyte itself so that it may serve some function. Keep in mind that these responses occur without any cognitive impact and are essentially reflexes. Basically, we aren't cognizant of the metabolic effects of gut taste, but these effects are important for regulating diet and metabolism. Endocrine responses may be triggered by nervous stimulation or by signal transduction within the enterocyte itself. Early work suggested that these responses may be paracrine in nature, where hormones trigger other nervous responses to signal nearby cells. But hormone secretion seems to act through endocrine secretion as well. This modality occurs when the secretory hormones are released into the bloodstream or lymphatic system. It is interesting to note that the greatest number of endocrine cells are indeed found in the gut, although they are dispersed throughout the gut, making them difficult to study. The phrase gut taste arose because of the discovery of receptors in the gut that are identical to the proteins on taste cells in the oral cavity. Clearly, these proteins do not share the same effects as their relatives in the oral cavity, since we do not receive sweet, sour, salty, umami, or bitter signals when our stomach contents empty into our intestines. Nevertheless, the term gut taste has caught on in the literature, regardless of its accuracy. The true function of these proteins in the gut is still being investigated, and while the receptors certainly seem to be sensing the environment of the gut, the term taste may be misleading. Now let's look into some more specific examples of the phenomenon of gut taste. While mRNA transcripts of T1R, T2R, and gusteucin proteins involved in taste have been found in a variety of intestinal and colonic cells, the functionality of these taste receptors in the gut is still unclear. One theory is that the umami and sweet receptors sensing nutrients in the intestine could be a mechanism for signaling satiation, leading to meal termination. Both gusteucin and the sweet receptor proteins T1R2 and T1R3 have been identified in enteroendocrine cells in the intestine responsible for the secretion of GLP-1. Gusteucin has also been found in cells secreting PYY, and the sweet receptor has been found in cells secreting GIP. These hormones are often considered anorexigenic signals in humans, so it's possible that sensors such as the sweet or umami taste receptors could detect nutrients in the intestine and lead to post-ingested feedback indicating fullness. Another group found that the transcripts of the sweet receptor T1R2 and T1R3 were found in greater concentration in cells at the tip of intestinal villi than in the crypts between the villi, indicating that these receptors may play some role in enterocytes that are actively absorbing nutrients from the intestinal lumen. Indeed, some research indicates that treating intestinal cells with sweet stimuli can lead to the upregulation of the sodium-dependent glucose transporter. This has led to the hypothesis that exposure to non-nutritive sweeteners could lead to differential glucose absorption. In mice, feeding non-nutritive sweeteners in conjunction with glucose did lead to a faster absorption of glucose into the bloodstream. However, this result was not found in humans. Thus, more research is needed to understand the role of sweet receptor activation in the human intestine. Bitter receptors have also been found in the human stomach, small intestines, and colon. In animal models, activation of T2R receptors leads to slower gastric emptying and an increase in ghrelin secretion, but these results have not been consistent in human studies. In the small intestine and colon, the bitter receptors may play a role in detecting toxins, just as they can detect toxins in the oral cavity. In cell culture studies, T2R receptors, sensing bitter compounds, increased expression of ABC transporters, which are responsible for effluxing toxins out of the enterocyte and back into the lumen for excretion. In studies using human and rat intestinal tissue, treatment with bitter compounds increased anion secretion by the tissue. Clearly, further research is required to understand the role of taste receptors found in the gut. At Purdue's Ingestive Behavior Research Center, or IBRIC, advanced and diverse research facilities are available for students working with gut taste, as well as many other topics pertaining to ingestive behavior.